So let's just go out of the world of definitions and talk about a phenomena which is more like multicellularity, right? I mean, in that case, it's not like we are talking about, again, single cell or a specific thing, but it's more like a continuous system, as you described.
It's the
So
exact same issue, right?
yeah.
At what point does something which forms groups become a multicellular organism? Or is it multicellular, which implies that it's an adjective modifying organism? And then you have to know what an organism is too. So yeah, it's the same problem.
Yeah. But then what is a multicellular system? At least, can
Yeah,
you
so
just like,
that word, I know I'm talking a lot, so I'll be fast, but that word has many different meanings depending on the community. It can mean it's simply there's more than one cell. It can mean that it's a Darwinian system such that the group of cells is evolving as an entity, or it can mean that it's an organism where it has functional integration of parts and the lower level units don't do that well on their own anymore. So, most of the time, those three things are actually all met. For something like an animal or a plant, um, they are multicellular. There's more than one cell. They're Darwinian at the group level, and they have functional integration. So they're organisms. But we often don't differentiate between those three states. We just say the word, and then it can be very confusing. So the way that I like to think about this is that the evolution, so like multicellularity, that word really encompasses, we care about all three. We care about how groups form. We care about how groups become capable of evolving novel group level properties through a Darwinian process, because it's that process of adaptation acting on group level traits that's injecting information into groups over generations and making them go from being just a simple clump to something which has behavior and morphology and the things that we ultimately care about. We wouldn't care about multicellularity if it was just undifferentiated blobs of goo. Like, we care about them because they can do things single cells can't. And then ultimately, the phrase that we use is that they are organisms. And that means that they are actually, they do display functional integration at the lower level, which gives rise to novel properties and traits at the group level. And similarly, that means that the lower level things are now parts of that new organism. And what's one definition of parts that I like is would be like the counterfactual. If I took that part and pulled it out of the organism and put it in the environment, how would it do? And if it's no longer doing very well anymore, if it can't go back to being free living very, very easily, then it's now this integrated, interdependent part of the new organism. And at that point, I think everyone agrees that that new entity that forms groups, those groups are Darwinian, and they've evolved to become a integrated whole, is a multicellular organism.
well, in this case, what I think is that there is also some sort of goal directedness. There is some sort of level acting because of the niche where you end up. For example, the work what Mike did, with xenobots, simply taking the skin cells of an embryo and just putting them in an individual environment, which it just forms like a group of cells, but they act like as an individual, right, Mike? So in that case, that already kind of highlights that this doesn't become like it has its own instructions in a way, right, which it follows.
So we could talk about Xenobots, I think, in a slightly separate context. One thing that I wanted to sort of mention this into what you and Will were just talking about is Physarum. So these slime molds, right? So this is something I really love, because this thing it breaks a lot of these definitions, right? And so it's supposedly unicellular, it doesn't have boundaries within it, but the thing can be any size, so you can have this giant thing. It has lots and lots of nuclei. Not super clear how much differentiation there is within it in terms of what almost certainly it has some, but it hasn't been super well studied in terms of how the different parts of it are, let's say, physiologically, molecularly different. But you can do all kinds of things. You can cut it into pieces, and the pieces do perfectly well on their own, but you can also they will also join. The thing can learn, and if it does learn, then you sort of face it next to another one that is naive. The information as they merge, right? The information, so this is Audrey de Saussures' work. The information will actually get propagated across across the whole new individual. So this thing can sort of scale up or scale down, sort of horizontally. And you can do interesting things like, for example, if you have a FICE arm and you put a piece of oat at a certain distance, and the FICE arm, starts going for the oat. Oh, by the way, another wild thing about it is that in FICE arm, its behavior and its morphogenesis are happening in the same space. So unlike many other creatures where you've got behavior in three-dimensional space, you've got morphogenesis. Its behavior is changes in morphogenesis. That literally is how it operates in the 3D world. So what you can do is, as this thing starts going for the oat, you can take a razor blade and cut off the leading part of it. And now something very interesting happens, right? Because now it's got a problem. It's got a problem to solve. That leading edge can do one of two things. It can go and get the oat, and if it does, it's huge nutrient density. It doesn't have to share it with the rest of the mass, so that's cool. Or it could go back and it could rejoin the rest of the mass.
I love it.
Here's
I love it. What are you gonna do?
What are you going to do? Because this is so funny. This is actually I was doing a home study unit with my oldest son, and he actually suggested this experiment, and we did it. And the other thing you can do is you can rejoin the other mass first. But something that's very cool is that payoff matrix that you have, as far as here are my actions and here's the payoff I'm going to get. The payoff matrix changes as a function of each move, because as soon as you rejoin the mass, that whole question of I'm going to go get it and not share it with the rest, that goes away. That doesn't exist anymore, because there is no me and you, there's just us. And so, right. And so this kind of like plastic allows a really weird kind of game theory where the actual payoff table, the number of players in the payoff table can change,
Mike, what's the genetic relatedness of the lower level chromosomes? I guess they would be, gets eukaryotes, so lower level nuclei, across within the syncytial body of the Physarum. Are they all clonal, or are there a bunch? Is it, highly polygenic?
Yeah. So they definitely can be clonal, and whether all of the wild ones are or aren't, I don't know, because who knows how much of this merging happens in the forest floor. Like, I don't know, but definitely, if you wanted to make a clone, like typical lab cultures are clonal, because you get a tiny sclerotia and you sort of expand it out into like meters of this stuff. So typically they're clonal. But we say our planaria are clonal too, but that's not really true. And even the clonal ones, their behavior is completely different. Their personalities are totally different, and you learn that when you try to train them. You find out that shoot, even the clonal ones all do whatever they want. So I don't know. I don't know how much of that is driven like directly by their relatedness or physiological history or what.
And by the way, this is a total aside, but I was watching the HBO hit like Rome show from the late 90s, a few years back. And in that show, one of the soldiers is returning back after Caesar's, crossing the Rubicon and coming back to challenge whoever his rival at court was, Pompey or someone. And one of his soldiers steals the loot train and goes off on takes the loot train for himself and goes off on a drunken binge, and then later gets caught by his own troops, by Caesar's troops. And he's like, oh no, I was just keeping it safe for you guys. And Caesar's like, well, I could kill you, but I think I shouldn't bite the hand of fate. I'll give you 100 talents and get out of here. It's maybe think of exactly the situation. It's separated from the main army. It finds the food. Now it has a choice.
Yeah. By the way, it looks like, and this is we definitely need more data, but it looks like it prefers to rejoin first. So not all the time, but the majority of them will first go backwards, join the collective, and then go get it. Some percentage of them don't, but mostly they do.
So cool. I mean, honestly, I think that the behavior of these things which are doing what multicellular life does, but they don't have cellular boundaries. Physarum being one, these macroalgae like Caulerpa, which are single-celled but meter scale algae, which is actually an invasive species in the Mediterranean, but it has stem and root-like rhizoids and frond-like leaves, but it's all one cell with billions of nuclei in the same way that Physarum has. You kind of have to maintain a constant nucleus to cytoplasmic ratio so that you can make transcripts that are used locally. But presumably, and I don't know the details here, but I think there's basically nuclear specialization in different tissues where the gene expression is different. That gives rise to different structures. But if it weren't for these cool examples, things like Physarum, things like Caulerpa, I would have probably argued that multicellularity is the best and maybe only way to coordinate large scale morphogenesis. Once you're talking about meter scale biology, just because single cells, you need some way of creating gradients and specializing. But that's clearly not true. And I think it's really effective at that. But these counterexamples show that you can certainly do macro scale complex organismal biology in the absence of cellular boundaries.
There's another cool example, which is acetabularia. So these guys probably about 10 centimeters, tall, and one nucleus. So they got a root, they got a stalk, and then a cap, like this beautiful cap thing. They live in the ocean, but just one nucleus. The whole thing is one.
So are they doing like what protists do with a macronucleus and like some sort of lower level generals of micro? Well, they have a nucleus and a macronucleus. The micronucleus is their sort of germline. The macronucleus is being used. Do they do something like that where there's like some way of like doing local local transcript exponentiation or something?
I don't know. There was somebody years ago that whole system to be molecular, and I don't know that ever happened. But but it's too bad. I think it's really important that.
So we were talking about, like, for example, why unicellular, multicellular, organisms, or the prokaryotic multicellular organism, something like organism, let's say, will not be able to communicate, let's say, in the long distances, etc. Than eukaryotic multicellular organisms. So you said that if there wouldn't be these few examples, you wouldn't like go with that, or you wouldn't kind of really, you wouldn't think that this is possible, right?
Yeah, to be clear, I was not this isn't so much a prokaryote eukaryote divide, but is a single bag of cellularized biomass a good substrate for getting something that has spatially differentiated tissues, or do you kind of need cells to be able to have discrete behaviors, right? Yeah. And clearly multicellularity has evolved many times, and it's an incredibly successful way to build differentiated bodies. And I think had we not had these examples where single cells, so you have these basically people call them syncytial groups, but basically you have a giant bag of cellular biomass without discrete boundaries around individual nuclei, and you might have millions or even billions of distinct nuclei, and you can have organisms that seem quite sophisticated. So you can have seaweeds that have roots and stems and leaves. They're not really those things, but they look just like them, and they function that way. You can have Physarum, a slime mold, which is probably the most similar animal-like behavior outside of the animals. It crawls around at relatively fast speeds through leaf and wet surfaces of forests, and is foraging for microbes to digest. And its whole body is moving around, and it's an active modal predator. And you don't see those things outside of animals very often that are big. You have plenty of single-cell protists, which can be fascinatingly complex, but they're like millimeters scale, and this thing can be 10 centimeter scale or bigger. It's kind of no real limit as far as I know on what their size is. And then Mike actually brought up and I was saying, well, I would have thought that multicellularity is necessary, and we know it's not because of these incredible organisms, but they still have a kind of roughly stoichiometric ratio of nuclear material to cellular cytoplasm. So these things that are big accomplish it by having many, many different copies of their genome, which raises all sorts of really interesting biological questions. But one of the things that they do is that a local genome can coordinate its own local behavior, and you could still have some sort of gradient, a biophysical gradient or a signaling gradient, allowing for cells for nuclear specialization, not cell specialization because they don't have them, but for a nucleus in a leaf tissue to do something different from a nucleus in a root tissue, right? No problem. That makes sense.